So I'm going to assume that you guys are all familiar with the cell theory. You've probably heard it since you were about 12 years old. It's the idea that all living cells must come from other living cells, which is what this little GIF here is going to show us. And there's basically three components to the cell theory. The first, which I just said, cells come from other cells. Two, cells are the smallest unit of life, meaning that living things cannot be any smaller than one cell. That's the basic unit of life. And the third is that all living things are in fact made from cells. And so we call that part of the cell theory, that all living things are made from cells. Careful, that's not one of the 7 functions of life. That's just another feature of life. It's part of the cell theory, that all living things are made up of cells. So one of the understandings from our topic 1.1 is just understanding the cell theory. Just being able to regurgitate these facts. Living cells are composed of cells, cells come from pre-existing cells, and cells are the smallest unit of life. Now, in this class, what we're going to do repeatedly is take these understandings from the syllabus and then connect them to something a bit more interesting. We call these the applications, and they're listed in your syllabus. That's posted in Canvas at the beginning of this module. So the application here for the cell theory is taking strange cells that would challenge the idea of the cell theory to see if it still holds up. So here's one example. The first example that you need to know, they're called striated muscle fibers, and we're looking at a few right here. So this long snake-like thing looks like it's scaly. That's a muscle fiber, and that's a single cell. And these little stripes are called striations, and that's where the name striated muscle fiber comes from. So what we're looking at here is an atypical cell. Now, why is it atypical? Because it's huge. It's not like most other cells. It can be up to 20 centimeters long in some cases for a single cell, which is very large for one cell. So that's unusual. Two, it's actually a unique type of cell that is a fusion of multiple cells, which sounds strange. Let me explain. When you're developing as a little embryo, the stem cells that will make up your muscle tissue, what they do is they start to fuse together. So they start out as separate cells, and then they come together in this bundle, which makes up one larger cell. And that's unusual for a cell. Those stem cells that make the fibers, those are called myoblasts. And then the third unusual thing is that because it's a fusion of multiple cells, and because these cells have to do lots of protein synthesis, they're going to actually have multiple nuclei. The word for that is multinucleated. And in this photo here, all these little purple dots, those are different nuclei within one cell. All of these things are very strange, very unusual. But does it reject the cell theory? No, because living things are still made of cells, right? So these are still cells. Does not challenge the idea that cells are the smallest unit of life, even though some cells happen to be huge. And then it doesn't challenge the idea that cells come from pre-existing cells. So we're still in good shape. So the cell theory holds up even with this strange example. Now IB wants you to look at 3 different examples. These striated muscle fibers are the first, and now let's move on to the second, and this is referred to as sometimes just giant algae. Here's our photo of the giant algae. The scientific name, or scientific genus rather, the Acetabularia. Now these guys are, again, strange because they are huge, and if you look at them, you wouldn't think that this is just one cell. This is actually a single-celled organism. This fan-like structure is just an extension of the cell, and then at the base of one of those fans, you can see it stretches out, and it looks like a stem. So it looks like we're looking at a plant with stems and leaves, but in fact, these are just single-celled organisms, which are up to about 100 millimeters in length. Very strange, very unusual cells, but they do not reject the cell theory. So one thing you might want to note, well actually 2 things, that these giant algae, they challenge the cell theory because they're very large, and because they're highly differentiated to have different structures without adding multiple cells. So they're unusual because they have these highly developed structures like that fan and that stem, but they are still individual cells. So unusual cells, large, highly differentiated, but they do not challenge or do not reject the cell theory entirely. The third type of cell that we need to look at is part of a mushroom or fungus. Now, I don't know if you're aware of this, but some of the oldest creatures living on Earth are fungi. For example, there's an example of a honey fungus growing that is around 2,400 years old. So that's ancient. And basically what happens is the fungus, the part that you see, the mushroom cap that you see up here, that's only a small portion of the organism. Most of the living organism, most of the fungus, actually lives underground, and what's referred to as its hyphae. So if you look underground, these hyphae are like little fibers that stretch out, and they're going to release digestive juices onto stuff, onto dead stuff, and then digest it like wood, and break it down, so that then the mushroom or the fungus can then absorb all of that digested material. And so if we were going to focus in on just some of these hyphae, you're going to see two different varieties. Let me get this out of the way. So we have some fungal hyphae. Again, these are like, they look sort of like the roots of the fungus, but we don't want to call them roots. They're hyphae. They serve a different purpose. Again, these are digesting stuff for absorption. So some of them have little walls that divide their cells. So these are typical looking cells, no big deal. Over here, on the other hand, this is what's called a non-septate hyphae. So the third type of cell that you guys need to recognize that challenges the cell theory is non-septate fungal hyphae. And the reason they're unique is that this is all one continuous cell, because it's not divided into smaller cells. And what that means is that in some cases, this single cell can stretch out for over a meter. And in cases even longer, and they're not going to have any divisions between them. So you could have a single cell that could be, well, potentially bigger than me. So these non-septate fungal hyphae are very unusual cells, very large, and just like the striated muscle fibers, they also are going to be multinucleated, lots of nuclei. So those two features make these cells unique, and that challenges the cell theory, but it does not reject the cell theory. Now, so far we've been talking almost exclusively about individual cells, but as you're aware, life gets far more interesting as we move to multicellular creatures. Now, first of all, in the syllabus, there's an understanding that says multicellularity gives rise to emergent properties. I want to just explain that really quickly. Now, multicellularity, obviously we're talking about multiple cells in one organism. In order for all of those cells to work together, they have to start to perform a similar function. And so we call this process where adding new, unique functions, we call these emergent properties. So here's how it works. As you move from a single atom to a molecule, that molecule can do things that that atom could not do alone. So the molecule now has new emergent properties. As you move from a molecule up to, let's say, an organelle, that organelle, something like a nucleus, will have properties that were not possible with just some molecules. That organelle has new emergent properties. Moving up to a single cell, that will have new emergent properties that those organelles could not carry out on their own. As we move from organelles to cells, the same is true. From cells into multicellular organisms, now we have new emergent properties which have to do with those cells working together, like, for example, making an organ like your heart or your brain. Those can do things that a single cell could never do on their own. And so moving forward, we're going to talk about how cells can work together to turn into special tissue types and carry out their new emergent properties. And this all has to do with the very first few cells that you're born with, which are called your stem cells. We're looking at my first baby photo. I'm adorable, right? Look at me. Look at this. This is just adorable. Check me out. Okay, so what we're looking at here, I don't know who this is. It's not me. We're looking at a zygote. Now, a zygote is a name given to the very first cell, which then develops into a multicellular organism. And a zygote is the fusion of the nuclei from two different parents. And what you wind up with is a single cell, which then has DNA from two different parents. This usually only happens during sexual reproduction, where in humans, for example, the sperm cell meets up with the egg cell, and then the nucleus from the sperm enters the egg, and now we have a fertilized egg cell. Now that winds up becoming a zygote. And then that zygote is going to divide through a process called mitosis, which you've heard of, I'm sure. And through that division, this single cell will produce all of the cells which become your bodies. So here's the hang-up. Now, if you look at the zygote, it's a single cell. Again, yeah, it had DNA from the two parents, but then that's it. So how does one cell with one set of genetic instructions decide to turn into eye cells and skin cells and, well, eye cells is misleading because your eye actually has several types of cells within it. Retinal cells, photorecepting cells. How do they know what to do? Well, that's the story of how your stem cells begin to differentiate, and that's our focus here. Quick vocab term for you. This word somatic. You need to get familiar with this word somatic. Now, in biology, the word somatic refers to body. Soma, that prefix soma, means body. And so your somatic cells, those are all of your cells throughout your body, anything that's not sperm or egg. So other than those two sex cells, anything else is referred to as your somatic cells. Now, every somatic cell in your body has exactly the same DNA, all the same genes, completely identical. So that if you were to take one cell, any one cell from your body, you would find that it has exactly the same genome as any other cell in your body. So if they all have the same instructions, all the same DNA and the same genes, how do they turn into different cell types? Well, let's start at the beginning. Your stem cells, at the very beginning of your life, the couple first cells that make you up, we call these your embryonic stem cells. They are completely undifferentiated. And what that means is that they still have the ability to turn into any type of cell. So they're going to divide quite a lot, and then they're going to start to differentiate into cell types. And so these early embryonic stem cells, and all stem cells actually, have this in common. They have the ability to differentiate into different cell types. Important. And two, all stem cells retain the ability to divide repeatedly, so they can make more and more cells. So if you want to make a bunch of skin tissue, for example, you can use stem cells which then divide repeatedly to make a whole bunch of tissue. Now, getting back to the embryonic stem cells, they have not begun the process of differentiation. So they have the potential to become any kind of cell they want. And so what happens is, as they develop, they're not going to use every single gene and every part of their DNA all at the same time. In fact, they're only going to start using the genes which are required for a particular cell type. And so depending on where that cell lands in the body, it will start to differentiate by expressing some genes, but not others. And so after time, as a cell starts to express some of their genes, but not others, it starts to become more and more like a certain cell type. And so skin cells will express proteins involved with skin cells. And so they start to become more and more skin cell-like, whereas nerve cells will start to express genes which are important for nerve cells. And that's how they, while they all have the same genes, your nerve cells and your skin cells and your hair cells all have the same genes, they're expressing some of them and not others, and that leads to differentiation. Now the next part is going to discuss how different types of stem cells exist and how they can differentiate and to what extent they can differentiate. Okay, so now let's talk about this concept of potency. Stem cells can be taken from embryos, like what you see here. After the zygote divides a couple of times, you can just snag those cells from the embryo and you can use those. And embryonic stem cells like those are the most potent. And what that means is that they have the ability to differentiate into many types of cells. The word for these types of cells is pluripotent, meaning that they're able to differentiate into basically any cell type. In this case, if it's a human embryo, then these could differentiate into any human cell type. And those are the embryonic stem cells. But there are other types of stem cells. Here's another example of a stem cell. Now, the umbilical cord, if you're not familiar with that, is a cord that connects a developing embryo and fetus to the placenta so that the mother can exchange nutrition with the baby as it develops inside. Now, the cord, the umbilical cord, will contain some stem cells in the blood that runs through it. And so these stem cells are not embryonic stem cells, but they are stem cells that kind of are moving around in that blood supply. And we can extract those stem cells. And they're great, but they cannot differentiate into any type. So they're not going to be pluripotent, these cord blood stem cells. Instead, the word we'll use is multipotent. So they can't make any cell type. They're not pluripotent, multipotent. And so that means that they can basically become a few varieties of blood cells, for example, but they're not going to make, some retinal cells for your eye. So that's cord blood cells. Now, these are still very useful, mostly because you don't have to create an embryo and destroy an embryo to get those cells. Embryonic stem cells require, as the name implies, an embryo. That's not needed here. Every time a baby is born, you can just extract some blood from that cord. It doesn't hurt anybody. They're throwing the cord in the trash anyway. Might as well take some of that tissue and use those stem cells for therapeutic reasons later on. In addition to that, that cord blood can be frozen and stored in a cell bank. So those are much easier to store than embryonic stem cells would be. So those are pretty ideal, except they have the downside of not being able to differentiate into any cell type. You also have stem cells in your body to this day. Perhaps you've heard of a bone marrow transplant. And during a bone marrow transplant, what you're after is these stem cells. We call these adult stem cells because they're still found in your adult tissues. One of the most common places for adult stem cells would be inside of your bones in this goopy stuff in the center called the bone marrow. Now, you might not know this, so in case you don't, your blood originates from your bones, actually, and it's the bone marrow that's responsible for producing new blood cells. And so those new blood cells will come from adult stem cells, and they have the ability to differentiate into different types of blood cells, white blood cells, red blood cells, and these little guys called platelets. And so what we can do is we can extract that bone marrow and we can access those stem cells for certain types of therapy. Think of treatment for leukemia, for example, or bone cancer. What we want to do is we want to eradicate all of the cancer cells and then replace those stem cells with new stem cells from a donor. So, like cord blood, these cells are not going to be pluripotent because they're not making any type of cell. We call these multipotent. They can differentiate into various blood cell types, but not any kind of cell. So you're not going to grow a whole new person out of this. You're not going to be able to replace eye cells necessarily, but you can easily make various types of blood cells. These are also easy to store, and so that makes them pretty beneficial, and they don't have the ethical downside of coming from an embryo. There are different issues with consent when it comes to cord blood and bone marrow. You have to find a willing donor that matches, but other than that, these are ideal sources for different types of therapy. Okay, so let's talk about how all this differentiation stuff actually happens. Normally, cells will grow together in little clumps when they're embryonic stem cells like this. So embryonic stem cells tend to stay together into clumps, which makes sense because you want the embryo to stay an embryo and not just break apart. However, if it does break apart, that's how you can get twins, but we'll talk about that later. So you get these little clumps of cells growing together, and then they're going to release chemicals into the environment around them. So imagine that your little cell growing next to your neighbor's cell, and this cell is going to release some chemicals, and that will encourage this cell to develop in a way that's similar to this cell. These chemicals, collectively speaking, can be referred to as transcription factors. What that means is it encourages those cells to express specific genes through a process called transcription. We'll talk about that later in the year. Today, if you understand that basically there are chemicals that cells release that encourage the expression of certain genes, then we're good to go. So if we manipulate the types of chemicals present, then, or in the ratio and the amount and the quantity and all that, then we can encourage these pluripotent stem cells to start to differentiate into a specific type of cell. And what's really interesting, actually, is that we can take multipotent adult stem cells and we can trick them to become induced pluripotent stem cells by kind of manipulating these chemicals again, and then we can get them back to act more like embryonic stem cells. And that can work sometimes as well. But right now, we're going to talk about therapeutic uses of stem cells. So, IB wants you to know one example of a therapeutic use of stem cells. What can we actually use stem cells to treat? And the example they want you to know is this condition called Stargardt disease. Stargardt disease, basically what happens is the retinal cells at the back of the eye, they develop, due to a faulty gene, they develop this sort of plaque fatty buildup. Now, what we're looking at in this picture is a photo taken through someone's pupil looking at the back of the eye. So you know the eye is just a big hollow ball, right? In the back of the eye, that's where the light hits. And this should all be filled with cells which detect the presence of light, and that's how you see these photorecepting cells. And there's one region in the back of the eye called the macula, and that macula starts to have issues in a human that has Stargardt disease. And you can see over here all of this macula atrophy. That's plaque. It's this fatty buildup that is a result of cells that have faulty genes. And since the genes are making this plaque, it's not like you can go in there and scrape it out. I mean, that would be probably really awful. But even if you succeeded, the cells will just continue making more of that problematic stuff. And so you have to get that faulty gene out of there. Now, how do you do it? Well, you replace that with stem cells, so that the stem cells that go into the eye will then go in and replace the cells that have the bad gene that causes Stargardt disease. Now here's how it works. Basically, you're going to start with some stem cells. You want to start to differentiate them into retinal light-detecting cells, like the ones found in the back of the eye. The difference is that the cells that you're adding here, they're not going to have that mutation that the person with Stargardt disease has. They're going to be healthy retinal cells. Then you take a needle and you're going to stick those embryonic stem cells right here, right as close as you can to the macula, so that they grow and hopefully grow at a healthier rate, and then start to replace the problematic cells that the original person had due to their Stargardt disease. And they've had really successful trials so far in treating Stargardt disease using stem cells, because those healthy stem cells will replace the faulty cells at the back of the eye. Okay, so now what you guys need to do for homework is you need to research some other therapy, anything you can get your hands on, and you need to tell me a little bit about that therapy. First of all, you have to outline the condition. What is the disease? For example, this is Stargardt's disease. You also need to discuss the source of the stem cells. Are they embryonic stem cells? Are they adult stem cells? Do they come from cord blood? Where do those stem cells come from? Where are they sourced? You need to identify the actual therapy. So it's not good enough to just say stem cells are used. How do they get them there? In this case, for Stargardt's, the healthy embryonic stem cells are injected into the eye. You need to describe that process as well. Certain diseases require chemotherapy or the removal of the old damaged disease cells, and so you need to go in to tell me how that actually occurs. All the details will be posted to your assignment on Canvas, but that's just a quick overview. And this concludes our part 2 of the lecture series. The rest of topic 1.1 will be covered in class, so there will still be gaps in your notes from 1.1, but this is everything that you're going to get from the online lectures. And that's all. I'll see you in class.